Okay, yo, so now we'll do questions for the cancer cells and Down syndrome. Alright, so straight away to question 10, page 40. Okay, first question they ask, explain the formation of tumor. So, as we mentioned just now, remember tumor are formed from cancer cells, and cancer cells are formed from normal cells, healthy cells. So whenever they ask you how tumor is formed, like I mentioned in the notes, you have to mention how does normal cell become cancer cells, and then how cancer cells becomes tumor. So this one, okay, I just show you the notes because the answer is exactly the same as the notes. Okay, ta-da, over here. Okay, so number one, you usually can write this one first. So you can say that when it's disruption to DNA that controls the cell cycle. So when the DNA, the nucleus of the cell, got problem, it cannot control the mitosis process properly anymore. That's why cell begin to divide by uncontrolled mitosis and form cancer cells. So from normal cells, I undergo uncontrolled mitosis, and all the cell produced are cancer cells. Okay, so because when one cell divides into many cells, all these cells are equally cacat punya DNA. So all of these are cancer cells. So the last point you just have to say, when all these cancer cells accumulate, I will then get a tumor. Okay, so that is tumor. Now, easy as that. Next question. One factor that caused the formation of tumor in lungs. So lungs is usually caused by smoking. Okay, so you can say smoking. You can say smoking cigarette. Okay, which contains the carcinogenic compounds. So remember, carcinogenic compounds are compounds that cause cancer. So smoking too much will most likely cause you lung cancer. Or you also can say point number one here. Okay, exposure to radiation also can, not necessarily have to write smoking cigarettes. So next one, they ask you 2 ways to prevent being exposed to the factor you name. So you can say do not smoke, of course. Okay, and you also can say avoid exposure to radiation. Okay, sorry about the dog. Now, next, explain how radiotherapy affects the cell cycle in cancer treatment. So this one during the lesson punya video I also got mentioned it. So radiotherapy is whereby they use radiation to kill the cancer cells. So how does radiation kill the cancer cells? So this is, let's say, a cancer cell. Okay, cancer cell also have nucleus, also have DNA. So when you use radioactive ray, okay, to like how to say, shoot it into your body, it will destroy the nucleus, destroy the DNA of the cancer cell. So how does it affect the cell cycle? Obviously also will disrupt the cell cycle, and then the whole cancer cell will die. But at the same time, these radioactive rays will also kill all your normal cells. That's why, as I mentioned to you, that's why cancer punya patient, why their skin is always like very dry, a lot of red dots. Okay, their mouth, nose, saliva, lips dry, hair all drop because their normal cells, body cells also all died. Okay, so number one, you can say that. How does radiotherapy? So we say radiation is used to destroy DNA of cancer cells. Okay, so we say hence killing the cancer cells. Okay, cannot. Alright, so just one more question we will do. So you go all the way to the last question behind at page 18. Okay, it's actually page 52. Question 18, page 52. Okay, so this question a bit of meiosis and then also a bit of Down syndrome. So at the beginning one is like last week punya lesson. Okay, they ask you, what is the type of cell division? So obviously when you see you have tetrad crossing over, okay, you have, see, all this means the variation in gametes. This is definitely meiosis. Okay, now what's the correct sequence of stages of the cell division? So first, to arrange these stages properly, right, you need to know how to differentiate meiosis 1 and meiosis 2 once I look at the cells over here. So very simple. Meiosis 1 and 2 punya difference, the first one is, right, in meiosis 1, you will have chromosome aligned as bivalent. Remember the couple, they are always a pair. So when you see a pair of homologous chromosome, that is definitely meiosis 1. And also remember, your sister chromatid do not separate. Okay, means that your chromosomes still have 4 chromatid attached to each other. They don't separate. Meiosis 2 is the opposite. So meiosis 2 is just exactly like mitosis. Okay, you don't have a bivalent, remember? And meiosis 2, your homologous chromosome already separate, like the couple already break up, remember? So you don't have homologous. And your sister chromatid will separate. Remember, from like this, your chromosome becomes 2 sister chromatids. Okay, so when you look at a cell, when you want to differentiate whether it's 1 or 2, these are the characteristics, the aspects that you should look at. Okay, for example, when I look at W, so I know, okay, remember the faces, promat. Okay, so when I see this, I know definitely it's telophase. Okay, the last stage already. So whether it's telophase 1 or 2, I look inside here, so I see my sister chromatids separate already. So that's definitely telophase 2. So this one, I see bivalent. I see them crossing over. I mean, crossing over already done. So this is definitely metaphase 1. Okay, so when they align at equatorial, I know it's metaphase. So how do you know is it 1 or 2? I see got bivalent. So it's 1. So this one, chromatid separate ma. Chromatid separate. So this is anaphase 2. This one is also telophase. So since this is telophase 2, this is obviously telophase 1. Alright. Now, the correct sequence right now, so definitely the first one is metaphase 1. It's X first. Then you have Z, Y, and W. Okay. What happens to the chromosome during stage X? During stage X, your your homologous chromosome align at the equatorial plane. Okay, homologous chromosome align at equatorial plane. Okay, so what happened to the chromosome after stage X, and why is this chromosomal behavior important? So right after metaphase 1 is anaphase 1. During anaphase 1, what happened? Remember, your homologous chromosome will move to opposite poles. So we say homologous chromosome moves to opposite poles. So why is this chromosomal behavior important? Okay, so during anaphase 1, the main importance of anaphase, right, 1 is actually so that your chromosome becomes haploid. Remember, during meiosis, you want to produce haploid gametes. So the main place where diploid becomes haploid in meiosis is during anaphase 1. Because here you can see at metaphase I have 1, 2, 3, 4, 5, 6 chromosomes. But during anaphase, remember, they move to opposite pole, they will separate. So 3 will come here, 3 will come here. So your chromosome number becomes half, becomes haploid. So you can say this is to produce gametes that are haploid. Okay. Now, next, name process P and Q. So you can see sperm and ovum combine to each other. That's the process that we learned before since last time. It's called fertilization. So when sperm and ovum undergo fertilization, what they form? They form a zygote. Okay, so Q is the zygote. Easy. Okay, now Down syndrome punya question. So remember, Down syndrome is whereby a person has extra chromosome in where? In the 21st pair. So these are the 23 pairs of chromosome in human. How many chromosomes are there in somatic cell of this individual? So since they have one extra, it is 47. What is the genetic disorder? The only genetic disorder we learn in this chapter is Down syndrome. What is the characteristics of karyotype of an individual who suffers from this genetic disorder? So when they ask karyotype, what's the meaning of karyotype? Karyotype means like this 23 pairs of chromosomes. That's called karyotype. So a normal person will have 23 pairs of chromosomes. A Down syndrome will have 23 pair, but an extra chromosome in 21st pair. So that is the characteristics you have to mention. So you can say has an extra chromosome in 21st pair. Okay. Now, what causes this genetic disorder? So why this person will have extra chromosome in 21st pair is because the gamete got extra chromosome. So why the gamete got extra chromosome is because, remember, during anaphase 1 or 2, what happens is your spindle fiber does not work. So during meiosis, during the production of sperm and ovum, right, the 21st pair punya chromosome doesn't split because your spindle fiber doesn't work. So you must say spindle fiber at 21st pair doesn't work, then your chromosome fail to separate. That's why I produce one cell with 2 of the chromosomes. Okay, and one cell kosong. So that's the first thing I explain. So number 1, I say that spindle fiber fails to function during anaphase 1 or 2 at 21st pair. So remember, you have to mention at 21st pair because the rest functions normally. Only this one got problem. Okay, so if the spindle fiber don't function properly, they will obviously cannot separate. That's why you can, next thing you must say, therefore chromosome fails to separate. So if chromosome fails to separate, what happens next is that one of your gamete will, like this, will have extra chromosome. So you can say, hence one of gamete produced has extra chromosome at 21st pair. So how do I get Down syndrome? Is when this gamete fertilized with a normal punya gamete, then I will have an extra chromosome, and then I will have Down syndrome. So I know it's already more than 2 points, but never mind, I give you the full answer in case they ask more points. So you just say lah, if this gamete fertilizes with a normal gamete, okay, then just look a Down syndrome child is produced. Okay, so yeah, that's how you answer the question. Ask how a person becomes or get a Down syndrome. So you remember, you have to start by saying spindle fiber fails to function during anaphase 1 or 2 at the 21st pair. So therefore, chromosome fails to separate. So this 2 is actually the main point. Okay, the other you just continue explaining. No? One gamete got extra chromosome, so normal chromosome fertilized with it, then you get the Down syndrome. Okay, so remember, after when you're done with this video, do the quiz. Quiz is just 10 objective questions. So if any questions, please ask me. Okay.